Okay, right now we're going to talk about the process of acute inflammation. Now, inflammation in general is a very high yield topic that you should totally understand clearly, and hopefully, this is what we're going to do today. Now, what is the definition of inflammation? Now, inflammation is a protective response of the tissue to a certain stimulus, for instance, to a foreign object or to necrotic tissue, or basically to a certain pathogen that infects that tissue. Now, we have two types of inflammation. We have the acute inflammation and we have chronic inflammation. Now, the major difference between acute and chronic inflammation is that the acute inflammation, of course, regardless of the timetable by which acute inflammation occurs and chronic inflammation occur. Now, the major difference is that the principal cells of acute inflammation are neutrophils. However, the principal cells of chronic inflammation are macrophages. Now, before I go into the details and tell you about the cardinal signs of inflammation, I should really focus and tell you a little bit about what mediates the acute inflammation. Now, we're going to talk about 5 different mediators. Okay. Now, let us suppose that this is a blood vessel. Okay, and this is the tissue outside of that blood vessel. This is the interstitium of the tissue. Okay. Now, suppose that there is a pathogen right here, and that pathogen invades that tissue. Now, we know that microbes or any pathogen, it expresses antigens on the surface. Okay? And we also know that within the tissue, we have cells of the innate immune system. For example, we have macrophages, we have dendritic cells, etc. Now, those cells, the cells of the innate immune system, they have certain receptors. And this is the first mediator. Now, this is called a Toll-like receptor, a TLR. Now, Toll-like receptors are of different kinds. For example, let's take an example. Let's suppose that this is a gram-negative bacteria. We know that the antigen presented on the surface is lipopolysaccharide. Okay, so the TLR, for instance, in this case, TLR CD14, okay, the Toll-like receptor CD14 can recognize that lipopolysaccharide and binds to it. Now, once it's bound, basically it sends signals to the nucleus to produce transcription factors. In this case, transcription factor, something that is called NF-κB, and this transcription factor will stimulate the cell to produce some immune mediators. Okay, and so this is basically the principle or the mechanism by which Toll-like receptors actually work. So this is the first thing that mediates acute inflammation. Now, we know that there are within the tissue that there are healthy cells right here, and when they sense the presence of an antigen, okay, now they will actually a certain enzyme will be activated in this case. Now, we know that the cell membrane is composed of a double layer phospholipid. So an enzyme is called phospholipase A2 will actually convert parts of the phospholipid membrane into arachidonic acid. Now, this is a really high yield point. Now, arachidonic acid. Now, arachidonic acid, it can go via 2 pathways, and arachidonic acid metabolites are really important in the process of acute inflammation. So it can either go with the path of cyclooxygenase enzyme, and this gives us prostaglandins, or it could go with the pathway of the lipooxygenase enzyme, and this will give us leukotrienes. Okay. Now, prostaglandins such as prostaglandin I2, prostaglandin D2, or prostaglandin E2, they mediate inflammation by causing an increased vascular permeability, and they cause also vasodilatation. Okay? So this is now an important thing about prostaglandin E2 is that it also mediates fever and pain. So just keep that in mind for future reference. We'll be talking about pain in the next video. Now, the leukotrienes, we have different types of leukotrienes. We have leukotriene C4, we have leukotriene D4, we have leukotriene E4, and we have leukotriene B4. Now those three, C4, D4, and E4, they also mediate increased vascular permeability. Now, you remember that with the normal vessel wall histology, we have endothelial cells, and just beneath that endothelial cells are different types of cells that are called pericytes. Now remember, the pericytes are found in the postcapillary venule. Okay, so when they constrict, it actually constricts. They actually constrict those pericytes, and they cause the increased vascular permeability. Okay. Now the leukotriene B4. Actually, you should remember that this is an attractant, so it is a substance that attracts neutrophils. Now remember something that is really high yield and important. Neutrophils are attracted by 4 factors: leukotriene B4, C5a from the complement system, interleukin 8, and bacterial products. Okay? Now these are really high yield points that you should really focus on and understand, because these are high yield for examination. Now, the next thing that we're going to talk about is going to be mast cells. Now, mast cells are also found within the tissue. Okay? Now, mast cells, as we know, they contain histamine granules, right? And when that histamine gets out, it causes vasodilation and increased vascular permeability. Okay? So, the mast cells are actually dormant, they're not activated. However, there are 3 things that can activate the mast cells. The first is being tissue trauma. So let's say that this is the pathogen and it releases some chemicals. So this is a chemical trauma, a chemical injury that will activate the mast cells. A photosensitivity problem. This is, or a temperature problem, a heat trauma. Any kind of injury to the tissue will activate the mast cells. Now, another thing that can activate mast cells is by binding of that antigen. Of course, they have IgE on their surface, and when this antigen comes and binds those IgE, it will activate the mast cells. The third thing is the C5a from the complement system. You should go and watch the video of the complement system, and you will see that the C5a actually plays an important role in the activation of the acute inflammation. I'll continue in the next video.